Thank you, Ken, for that very kind introduction, and I'd like to thank LINK-J for setting this up, and to thank you all for your wonderful hospitality. This is the first time I've been in Tokyo, and it's certainly wonderful to see what's going on with the research enterprise here, as well as to get a look both at the academic side and at the industrial side, and at the wonderful partnerships between them. So what I'm going to tell you about this afternoon is work we're doing on our dynamic microbiomes in gut health, and a lot of the stuff is so new it just came off the DNA sequencer in the last few months. So microbiome research is really changing how we view ourselves as humans, and I'd like you to take just a moment to consider, what did you see when you looked in the mirror this morning? I saw an organism that is just 43% human. And not just because I was jet lagged and it was early and I had not yet had my coffee. But when we think of what makes us up as human beings, each of us has about 30 trillion human cells, but about 39 trillion microbial cells. So that's where that 43% human number comes from. It is amazing to think that we are more microbial than we are human in terms of cell count. Now you might be thinking, it's the 21st century. Why do we care about counting our cells instead of thinking about it on the DNA level? So let's think about that for a moment. Each of us has about 20,000 human genes, depending on what exactly you want to count as a gene. But the size of our microbial gene catalogue ranges from 2 to 20 million microbial genes. And so by that measure, we're at best 1% human. And especially in terms of systems biology and systems medicine, which are getting more and more appealing at the moment, it is hard to do systems anything when we are neglecting 99% of the system, which is what we do when we disregard all those microbial genes. But perhaps most fascinatingly, the microbial genes that we have, which make up the vast majority of genes encoding the metabolic reactions that we can perform, those microbial genes are the genes that we can change and that we can transform. And as I'll show you, we have changed them profoundly throughout our lifetimes, and we continue to change them through actions that we have control over. And this is very much a big data problem, so it is amazing to think that each teaspoon of your stool contains, encoded in its microbial DNA, literally the amount of information it would take 1 ton of DVDs to store. So you can think about that the next time you're taking a dump. You are also taking a data dump, and much of that data, in fact all of it for most people, is just being flushed away at the moment, rather than used to improve our health. So in the Human Microbiome Project, which I was involved in several capacities, together with about 400 researchers around the US, we spent $173 million of NIH's money to characterize what does a healthy human microbiome look like, or at least that was the largest single component of the project. So in the paper in 2012 that came out in Nature, we looked at about 250 people at up to 18 sites in the body and collected 4.5 trillion bases of DNA, so 1,500 times the size of the human genome, only with microbial genes rather than human ones. And the great thing about this is that we had an unprecedented amount of DNA sequence data about the microbiome. But that was also the terrible thing about the project. And to illustrate this, I'm showing you the first file of data from the Human Microbiome Project. And despite the fact that what we are trying to do is ecology, it is pretty hard to tell who lives where in the environment from this sort of information, right? And what they interact with and how abundant they are. You probably can't even tell that that's an oral sample, let alone the sequence signatures in it that let us make that determination. And the worst part is that this is just the first 0.1% of this file, and there are another 17,000 files just like it. This is really an issue today because now, through crowdsourced projects like American Gut, that I will tell you about later, and also various companies, there are various ways where you, as an interested member of the public, can get microbiome sequence data about yourself. And so this is a huge problem for physicians right now because their patients come in with a huge grin, always with a huge grin on their face, and say, you know, doctor, I have some wonderful news for you, which is that I had my microbiome done, and now I have a list of 1,000 species that they found in my gut. And surely with all this data, you can tell me what's wrong with me, right? And what is your doctor going to do? Maybe they can refer you to their colleagues in psychiatry for being crazy enough to think that they could do something with that. And what our goal is, our challenge as a field, is to make it not crazy anymore and to understand from one profile, or perhaps a series of profiles over time, what that microbiome data has to do with a person's health and how we can use it to improve their health going forward. So back in 2008, we developed a pipeline for doing highly multiplexed assays, which is now widely used today, where essentially we barcode each one of hundreds of samples and then run them together on a sequencing instrument, initially 454, but more recently Illumina. And we developed a software pipeline called QIME, which stands for Quantitative Insights into Microbial Ecology, which is free, it's open source. It runs everywhere from your laptop to the $26 million Comet supercomputer in the San Diego Supercomputer Center at UCSD to the Amazon EC2 cloud. And essentially what it does is it takes the data right off the sequencer and turns it into more interpretable results that you can use and that you can publish. And when we apply this tool to the Human Microbiome Project dataset, we get this much more interpretable result, which is a map of how all these different microbiomes are arranged. So each point on this map represents all the complexity of a microbiome read out from its DNA. And we place 2 points close together on the map if they are more similar in terms of their microbiomes, in terms of their evolutionary history. And we place them further apart on this map if they are more different from one another, using a distance metric that we developed in 2005 called Unifrag. So you could imagine all of the different ways that a microbiome could be affected by traits of the person. What I'm doing here is coloring that data by the most important thing that relates all those samples in the Human Microbiome Project to one another. And on this map, almost like different continents, you see emerging the different parts of the body. So the mouth, the skin, the vaginal, and the fecal communities are radically different from one another. And to illustrate this, I highlight the mouth and the gut of the first person in the Human Microbiome Project, where you can see that those samples are in completely different places. But it was not until we did the Earth Microbiome Project that we truly understood how profound these differences in different parts of the human body were. Because in the Earth Microbiome Project, which was published in Nature last year, we crowdsourced tens of thousands of samples from different researchers around the world and ran them all through the same sequencing pipeline. And so what we could do is we could compare any two samples from the human body to samples out on the planet's surface and ask what samples were just as different from one another as these two samples from the mouth and the gut of the same person. So if you think of your mouth as being a little like a coral reef, where you have these complex mineralized structures covered with biofilms that perhaps your dentist pesters you about from time to time, the amazing fact is that the microbes in your mouth are as far from those in your gut in terms of their composition as the microbes in the water of this reef are from the microbes in the dirt of the soil. And that's amazing, right? Because it means that less than a meter along the length of your body makes more of a difference to your microbes than thousands of kilometers across the Earth's surface. And we never expected that, yet having microbial ecology as a common measuring stick to compare any samples from the human body or from the environment or anywhere else on Earth really lets us draw these astonishing conclusions. So shortly after I got to UCSD, I met Larry Smarr, who's the director of CalIT2, our California Institute for Telecommunications and Information Technology. And what I'm doing here is showing him the Human Microbiome Project data, as I showed you, but on his 64 million pixel high-resolution display wall. And Larry said to me, you know, Rob, I have a very interesting microbiome myself because I'm a patient for inflammatory bowel disease, and how do I get myself onto this sort of map? So not too long after that, this box arrived in my lab containing Larry's own samples. And Larry is a very interesting character because he's a leader in the quantified self movement, and he has been measuring things about his own body for the last 18 years, starting with one dimension of his weight, shown here, and then dozens of dimensions with blood tests and stool tests, then thousands of dimensions with human SNP profiling, and now millions of dimensions with sequencing the microbiome. So Larry had been sequencing his microbiome for a while before I showed up, and the general result is this sort of picture. And don't worry if you're not getting anything out of this in terms of obvious patterns. So basically the different colors are different kinds of microbes here at the family level, because Larry also did not get a lot out of this kind of information, despite spending, I don't know, a huge amount of personnel effort and compute time in getting this kind of information together. And the problem is that just having the microbiome data is not enough. You have to view it in the right ways with visualizations like the principal coordinates analysis map that I showed you to truly make sense of it. So we took the same data and ran it through QIIME, and what we get is this much more interpretable pattern where you see very clearly a red cluster and a blue cluster. And when we animate this through time, connecting up each point in his journey through the map, you see he starts at one edge of the blue, travels directionally through the blue, switches to the red, and then bounces around at random in the red. So you may wonder, well, what does this have to do with Larry's health? And so because Larry collected all that clinical information, we can match them up to each other. And what we see is remarkable. We see this initial shift caused by antibiotics, and then that directional movement through the blue, that is correlated with a loss of weight and frequent inflammatory bowel disease symptoms, during which his life is not very good. He then changes his medications, and that is what switches him from the blue region to the red region. And then in that red region, where apparently he was moving around at random directionally, what you can see is his weight goes back up to a healthy set point and then stabilizes. And what is remarkable about this is had we known about Larry's own configuration on the microbiome map, we could have told him as soon as you get into that red region, your weight will go back and stabilize to a healthy set point, and you will be fine for months after that. And so having this fine-grained ability to predict for an individual what their configuration space is to make predictions about what is going to happen to them with future treatment is a very exciting direction right now. And you can only do this with this kind of high-resolution time series data. So you might think that's all very well for Larry, but can we do this for more than just one person? And so with Janet Jansen and a number of other collaborators, we expanded these results to a much larger cohort in inflammatory bowel disease. And what I'm going to show you here is an animation for different people, colored by different clinical state. The two main groups to keep account of are the healthy people, who are in green, and then the ileal Crohn's patients, who are in red and yellow. And what you can see when I start the animation going is a tremendous amount of motion in those ileal Crohn's patients, whereas the people who are healthy stay relatively the same on this map. And so what is interesting about this is it is not just a snapshot of your microbiome right now that is different between health and disease, but rather the movie of how your microbiome changes over time. And these differences in stability, which we never knew about until we measured the same people over time, seem to be very important. Now that's just an animation, but how can we quantify this? Well, the answer provided by Yoshiki Vasquez, a very talented computer science grad student in the lab, was if we fit a plane as a mathematical model to the healthy people, then we can measure the distance of each point when the distance of each point, whether it's healthy or unhealthy, to that plane. So the top row shows you a cartoon of what is going on, and the bottom row shows you the fit of the mathematical model to the real data in this cohort. And what is amazing about this is that by using the dynamic properties of the microbiome, not just a single snapshot, we can get a better readout of disease than calprotectin assays, which were the current gold standard for reading inflammation out of a stool sample. And so we think these dynamics of the microbiome will be very important for a wide range of diagnostic applications. So although Larry was fine for over a year after the piece that I showed you, unfortunately he later had his inflammation come back and had to have surgery. So initially he had a colon cleanse, and what you can see here on the y-axis is the amount of phylogenetic diversity, so a measure of microbiome diversity. And you can see that it about halves after the colonoscopy, which interested his gastroenterologist a lot because no one had done a detailed daily measurement of how much does the colon cleanse affect your microbiome. When we do the principal coordinates map, what you can see here is that the variation after his colonoscopy is very large, and he goes to a completely different place than before the colonoscopy, which are those points in blue. But you can also see that within a couple of weeks, he has basically come back to having the same state as he had before the colonoscopy. So a lot of different taxa change. I won't go into the details of this, but it just shows you that when you have dense time series data, you can interpret a lot more about the relative abundance of individual kinds of microbes than if you just have before and after sampling. And what is most important is that when you have very high resolution data collected every day, you have more similarity one day to the next. So you can see the subtle shifts one day to another, and then also the much larger shifts, such as the one that you can see straight after surgery, where his microbiome has gone into a completely different configuration. So let's talk about that surgery a little more. What is amazing is that unlike the colonoscopy, after the surgery, his microbiome diversity goes down to less than a quarter of what it was before. And instead of recovering back to the state that it was before, it looks like it has not recovered yet, even several weeks later. But what about in principal coordinate space? Well, when I set this going, what you are going to see initially in orange is the samples from before his colonoscopy. And then you see it switches over to red after the colonoscopy, where he changes, but he comes back to the same state. Then there's a few more samples before his surgery, which are again in the same general area. And coming up next after the surgery, you see that huge shift, and then he starts to return. But instead of coming back to the same state like he did after the colonoscopy, you see that he has gone to a completely different state. So then you might wonder, well, does this difference in state actually matter? How big are these differences really? And to answer this, we integrated his samples with samples from the American gut population, which I will talk about more, but essentially these are crowdsourced samples from thousands of people around the United States. And so this data frame just quickly shows you the American gut population. And what you can see is you can see Larry's own samples that we can put into that data frame and use the information from many people to understand what's going on with this one patient. Okay, and so what we're going to do is we're going to bring that data frame back, where the brown samples are from the stool, the green from the mouth, the blue from the skin, and the pink ones from the vaginal community. And when we animate Larry through time, and this is exactly the same as the animation I just showed you, but now with these other samples from other people added, you can see that the difference with the colonoscopy is very small compared to the differences between different people. But when he has the surgery, which is coming up in just a moment, you can see the surgery shifts him so he is more different from himself the day after that surgery than the differences between any two people in the general population. And that is an amazing difference. And then you can see that when he's gone back to more or less the same configuration he was in before, but he looks like a different person. And so what's amazing about this is that the colonoscopy, although it caused quite a large change, it didn't make Larry look like a new person, in that he still came back to the same state, whereas the surgery has caused a permanent change in Larry's state that's larger than the difference between different individuals in the American Gut Project. And this is amazing because it's the first time that we've seen a permanent change in the microbiome from a medical intervention like surgery. So one really important question in linking the microbiome to health is whether controlling single pathogen species in the gut, which has led to some of the major public health successes of the 20th century, bringing all sorts of single organism infections ranging from measles to tuberculosis down to very low levels. Have those efforts to get rid of single pathogens led to a depletion of microbial diversity in our gut ecosystems? almost a silent spring situation, because as we have got infectious disease under control, there has been an explosion of many chronic diseases, including multiple sclerosis and Crohn's disease and type 1 diabetes and asthma. And what amazes me is that in 2002, when this paper that I took this graph from was published, none of those diseases were known to be linked to the microbiome, whereas today we know both from association studies in humans and from causal mechanistic studies in mice that not only are they linked to the microbiome, but the microbiome can be a causative agent. And there are many things that we are doing to deplete our microbiomes, everything from too much antibiotics to cutting ourselves off from healthy sources of natural microbes like the soil, livestock, the air, and so on. And my colleague Marty Blazer at NYU has written a wonderful book-length treatment of this in his book Missing Microbes, so I won't go too much into that here. Now, you may be wondering, does all this diversity in the microbiome really matter? And just over the last 10 years, in large part using the tools that I showed you before, the microbiome has been linked to a huge number of different diseases, ranging from ones you might expect, like inflammatory bowel disease and irritable bowel syndrome, to ones that are more surprising, like liver disease, rheumatoid arthritis, atherosclerosis, to ones that are truly surprising, like, for example, autism and Parkinson's disease. And one thing we have been studying for about the last 10 years with Jeff Gordon's lab at Washington University in St. Louis is links between the microbiome and obesity. And today, for example, I can tell you with 90% accuracy whether you're lean or obese solely based on the DNA from the microbes in your gut. So on the one hand, this is a cool trick from a technical perspective. On the other hand, you may be thinking, that does not have a lot of commercial potential as a test for obesity, right? Because I bet you can tell who is fat knowing nothing about their microbes. But on the other hand, if we try this classification task, lean versus obese, based on human genes, we can only do it with 57% accuracy using every gene ever linked to obesity by genome-wide association studies, whereas in contrast, we can do it with 90% accuracy based on the microbial genes. So then you may wonder, well, is it cause or is it effect? And to address this question, we move to notobiotic mouse models, where we start off with, where we take mice that have absolutely no microbes of their own, and then we infect them with microbes that we think will cause a phenotypic difference. And again, this is with Jeff Gordon's lab at Washington University in St. Louis. And what is amazing about this is you can take these germ-free mice raised in a bubble with no microbes and transplant into them the microbiome of an obese person. And what you get when you do that is an obese mouse. Now, fascinatingly, if you take that obese person's identical twin who is lean and you transplant that lean person's microbes into mice, then the mice remain lean. And so that is how we can prove that it is causal for obesity, at least in a mouse model, rather than simply being an association. So we were able to take this a step further with Ruth Lay and Tim Spector and a number of other collaborators in the Twins UK cohort, because a lot of people are interested in how human genetics may be related to the microbiome. And what we found is that monozygotic twins, so identical twins, are only very slightly more similar to each other than non-identical twins, although both types of twins are more similar to one another than to unrelated members of the population. But the effect is so small, you must look at hundreds of twin pairs to see it at all. However, that's at the whole microbiome level, and there are some individual microbes like the Christiansenella, where when you look at identical twins, so the monozygotic twins, they are highly correlated in members of the twin pair. Whereas when you look at non-identical twins, you see much less or no correlation. And so when we look at the heritable component of the microbiome, it is more likely to be biologically relevant. And in fact, this particular microbe is correlated with being lean in the UK population. So Ruth wondered what would happen if you took that microbe identified in this cohort study as being associated with leanness, and so she stuck it into mice that were going to become fat because they had received the microbiome from a fat individual. And sure enough, adding this microbiome associated with low body mass in humans also slimmed down the mice. So what is exciting about this is we can identify from a population study microbes that are able to change the phenotype of a mouse and slim it down. We are trying to do that in humans at the moment. We cannot do it yet, but certainly the potential is tremendous if we are able to find the microbe that allows humans to lose weight, as we can already do for mice. In case you think this only affects diseases that are obviously related to the gut, like the nutrient intake that is related to obesity, with Sarkis Masmanian's lab at Caltech, we have shown that we can do the same thing for Parkinson's disease, where the microbes in the gut have a large impact on microglia in the brain. And also for multiple sclerosis, where we see what look like very subtle differences between controls and multiple sclerosis patients when we look at individual taxa, although many of these subtle differences are statistically significant. However, what is not subtle is that if the mice receive the microbiome from a multiple sclerosis patient, they develop EAE, which is a mouse model of multiple sclerosis, at much, much higher rates than if they remain germ-free, or if they get the microbiome from an individual who is healthy and who does not have multiple sclerosis. So this is all about reading out current state. Where we would like to push this is to ask, from a test transplanting your microbiome into mice, can we tell not just do you have Parkinson's or multiple sclerosis or any of these other microbiome-linked diseases right now, but will you develop it using the shorter lifespan of the mouse to accelerate these age-related diseases and read them out much faster to tell you if you're at risk of these diseases in the future? Now, when we think about the gut microbiome, one very important factor is diet. And this idea that you are what you eat is certainly not a new concept. So this painting is from the 1500s, for example. And Jeff Bland, who leads the Precision Lifestyle Medicine Institute, often likes to say that food is a language that speaks to our genes. And what he means by that is that our human genes are fixed at birth, but food can change their gene expression in a number of important ways related to health and to longevity. And the language of food is very much expressed in color. So everything from the red of the tomatoes to the orange of the carrots translates into particular compounds like lycopenoids, carotenoids, and so forth. And we are learning how these plant natural products have tremendous impact on our physiology and on our health over the long term. Now, California is known for its healthy food and healthy lifestyles, but regrettably, the source of food closest to my house in San Diego is this. And when I take my 6-year-old daughter there, she sees many bright colors. What is particularly ironic is that their motto is too much good stuff, whereas we might argue that the good stuff has been systematically stripped out and replaced with synthetic analogs that don't have the same physiological properties at all. Now, studies of the impact of individual food items have been very challenging, and this is one of the most impressive studies of its kind. So a group at Harvard tracked 120,000 people over the course of 20 years, measuring the effect of each food item in terms of its ability to promote weight gain or weight loss in this cohort of 120,000 people. And if you think about your stereotypes of what thin people eat and what fat people eat, many of those stereotypes were very much reinforced by the study at the whole population level. Because it will probably not surprise you which food was most associated with losing weight, which is this, yogurt. But the magnitude of that weight loss was relatively small, so each additional serving of yogurt results in a weight loss, each additional serving of yogurt per day results in a weight loss of less than 1 kilo per year. At the other end of the spectrum, it will probably not surprise you what food was most associated with weight gain, which is our friend the French fry, amusingly America's #1 source of vitamin C, because there is not much vitamin C in each fry, but if you eat a lot of them, that is where you'll get your major dietary source of it. However, again, the magnitude of the change is relatively small. So each serving of fries per day leads to a gain of weight of less than 1 kilo per year. And putting this information together, what it means is that if every single day you decided to virtuously forego the serving of fries that you were going to eat and instead eat a serving of yogurt, at the end of exercising that willpower 365 times in a row, the difference to your weight, if you're typical of the population average, will be just a little over 1 kilo, which is perhaps not very impressive given the amount of mental effort that you have to put in to be able to do that. On the other hand, the individual variation is tremendous, and that is why these studies are so difficult, because if you make that diet substitution, on average, you will lose about 1 kilo, but for some people, they will lose 20 kilos. Other people will gain, say, 19, and so when you look across the whole population, it all averages out. So if we could find the basis for this individual heterogeneity and tell people what they should do on an individual basis, it would be amazing. And one group in Israel did exactly that in this groundbreaking paper that was published at the end of 2015 from Aaron Alanav and Aaron Segal's lab, where what they did is they looked at 800 people and hooked a subset of them up to continuous glucose monitors and then fed them a defined sequence of dietary items so they could look at the impact of each food item on blood glucose over the course of 2 weeks in 800 people. And then they took a huge number of measurements from the microbiome, from the blood, through questionnaires, through physical measurements, and through food diaries, and fed them all into a machine learning model to ask how could they predict individual glycemic responses. And what was remarkable about this is when they took the data from 800 people and averaged the glycemic index for the glycemic response for each food item, they recaptured the glycemic index values that were published to within a few percent. But what they found was that individual people had totally different responses to the same food item. So for example, they had one subject whose blood glucose went haywire every time he ate tomatoes, which you would normally consider healthy. And when he cut tomatoes out of his diet, his blood glucose was much better under control. But one thing that was remarkable is that they found that a fairly large subset of the population were actually better off eating ice cream than eating rice in terms of their blood glucose response. And they found that all of the variation in that, as well as in glycemic index responses generally, was linked to the subject's microbiome. And so they were actually able to use the microbiome to design an isocaloric and macronutrient balanced good diet and bad diet for each one of the 100 subjects, and show that based on their microbiome, they could predict what food items were good or bad for that person, at least in terms of blood glucose control. They did not look at weight loss in that study. And so on learning this, a lot of people have two questions. The first question is there a test I can do to find out if I'm in the group of people who should eat rice or who should eat ice cream? And the answer is yes, but it only really works if you're Israeli right now, because it needs to be retrained for different populations with different diets, different microbiomes, and different host genetics. But then the second and more interesting question is, suppose I found out that I was in the group of people who should eat rice. Could I change my microbiome to get it into the state where it would be better for me to eat ice cream? And that kind of general principle of how can you change your microbiome in a defined direction is exactly the kind of thing we are working on at the moment, although it is a very challenging and unsolved problem in the field right now. There are a lot of things that change our microbiomes, but changing them in a particular direction is still a very difficult challenge. So to meet this challenge, back in 2012, we launched this project called American Gut, where unlike the Human Microbiome Project, where you could only participate if you happened to read the right ad and show up on site at Washington University in St. Louis or at Baylor College of Medicine in Texas, the idea with American Gut was to make it possible for anyone to participate. And so what we did is we used crowdsourcing and crowdfunding and citizen science to open the project up to anybody. So it's kind of like National Geographic's Genographic project, where we achieve scalability by having each person support the cost of adding themselves to the project so that we can continue doing it as long as people are interested in it. Of course, it turns out that not everyone wants to know what's in there. And these are middle school students touring our lab and learning that we are going to use lasers and robots to read out the DNA of the bacteria in their poop, all of which is literally true. And you can see a tremendous amount of response heterogeneity here. Despite what we're learning about the gut-brain axis, we do not yet know if this is linked to their microbiomes, but before too long we'll have the sample size to find out. Joking aside though, most citizen science projects are on the scale of a few $1,000s, but American Gut has already raised over $2 million, essentially all in $99 increments from members of the general public. And we've sequenced and released the data for free for over 10,000 samples so that anyone can use it. And so the idea is that any educator, any researcher, anyone at a company, anyone at a government agency can go to this large database and understand what a large sample of microbiomes looks like and what kind of heterogeneity you are going to expect when you look at microbiomes from a lot of people. And so on the scale, we're finding connections between the microbiome and all kinds of things you might not have guessed that it was involved in. And that certainly in 2012, it was crazy to guess that it would even be associated with. So we had endless challenges with our ethics board to be able to get the study approved. But for example, you might have expected that your microbiome changes as you age, which it does. But you might not have expected that it would vary with how many hours of sleep you get at night, which is something that's just as statistically significant a relationship. And with a study of thousands of people, what we can do is we can subsample from that data set and put together power curves to guide new studies to ask, if I tried to study the relationship between the microbiome and some factor, how many people per group would I need to look at to be able to reveal that relationship? And so here on the x-axis, we have the number of people per group you need to see a particular relationship. And on the y-axis, the statistical power of the test. So 0.8 means 80% chance to detect. 1 means 100% chance. And so for the stronger effects like age or inflammatory bowel disease or antibiotics, you need a few dozen people per group to detect. But we can also see much more subtler influences that you need hundreds or thousands of people to look at. So for example, we can tell whether you're male or female with a few hundred people per group. We can tell how much you sleep, how fat you are, how much you drink, even how much you exercise, or I should say how much you say you exercise, because remember, all this is self-reported data. So either we see an effect of exercising or an effect of lying about it. And we're doing some work with UCSD student athletics at the moment to try to figure out which it is, because their coaches say that they keep them honest. But one thing that is amazing is that the steepest curve on the left, that we only need about a dozen people per group to detect differences in, is the number of different kinds of plants that people eat. And what is amazing about that is that this lifestyle factor that you have control over makes a bigger impact on your microbiome than things you have no control over, like your age or your sex or whether you've had to take antibiotics. And so what's exciting about this is that we see some of these lifestyle factors having very large influences. And what we need to know now is can they counteract the negative influences of things like, for example, eating a poor diet or not getting enough exercise or the various influences associated with drugs or disease. So the most powerful case that we have that shows you why you should care about where you are on these microbiome maps is the case of Clostridium difficile infection, or C-Diff. And this is just the Human Microbiome Project data to reorient you on this map. And what we're going to look at is some work that we did with Alex Karutz and Mike Sadowski at the University of Minnesota. And C-Diff patients have a particularly nasty form of diarrhea that's often antibiotic resistant and acquired in the hospital. And this is a disease that kills 14,000 people a year in the United States alone, and its incidence is rapidly rising in Japan and other countries around the world as well. And when I show you the microbiomes of C. diff patients, what you can see is that they look nothing like their healthy stool microbiome. So those orange spikes are all stool samples, but they're absolutely nowhere in the region of the map like the healthy stool microbiome, which is down the bottom in brown. Now what's going to happen is that 4 of these patients are going to get a fecal transplant from one donor. And so you might wonder, at the scale of the whole microbiome, am I even going to see anything? Because even though their microbiomes are profoundly dysfunctional, they're still pretty complicated. And transplanting in a small amount of fecal material from somebody else might not be sufficient to restore all that natural complexity. So you might also be wondering, what is a fecal transplant exactly? And to illustrate this, here's our chair of gastroenterology, Bill Sanborn, about to deliver a fecal transplant to a patient using hospital-grade stool that he got from a nonprofit called Open Biome, because the FDA in the United States regulates stool as a drug. And what's interesting about this is that 5 years ago, he told Larry Smarr, who I showed you earlier, we will never do a fecal transplant at UCSD, whereas now he does about 3 of them himself a week, and there's tremendous patient demand for this. So why is there such patient demand? Well, what I'm going to show you here is an animation showing the journey of these patients through the microbiome map. And each frame in this is 1 day of their journey. So starting off in that dysfunctional state. And you might wonder, how much do they move towards the healthy state? And is that transition fast or slow? Is it smooth or is it jerky? Does it take them a long time to get there? Do they stay there once they've arrived? And so what I'm going to do is I'm going to set this going. And what you can see is that immediately, in just 2 or 3 days, all of their microbiomes go into the healthy state, and then they stay in that healthy state during months of follow-up. And what's amazing about that is in those first few days, all of their clinical symptoms vanish and they don't come back. And in fact, fecal transplant has been shown now in many different studies to be a remarkably effective therapy for recurrent C. diff infection. And the last direct comparison to antibiotics that I know of had to be stopped early, because the fecal transplant was over 90% effective, which is typical for fecal transplant for recurrent C. diff. And the antibiotics were only about 30% effective, which is also typical. And so it was considered unethical to continue withholding the fecal transplant from the patients on the antibiotic arm of the trial. So they all got the fecal transplant, and most of them recovered. So the challenge that faces us at the moment is to figure out, for what other diseases can we identify a problem with the microbiome and then guide the microbiome back into the healthy state using this kind of map of what good and bad configurations look like. And there are many different ways you could consider reshaping the microbiome, whether it's something that sounds as extreme as fecal transplant, or something that sounds as benign as diet, or something that's in between, like antibiotics, or other drugs that modify the microbiome, like metformin or proton pump inhibitors, or prebiotics or probiotics or symbiotics, or phage therapy, or any of the numerous other therapies that are starting to come online based around modifying the microbiome. And we need to, so basically in order to do this, what we need to do is, for the expanding number of diseases that have been linked to the microbiome in humans and then demonstrated to be causal for disease in animal studies, what we need to do is we need to find the good and the bad places of the map for all of these different diseases. So we can tell you something about where you are in terms of your health based on where you are on this microbiome map. But we need to do more than this. So although we're pretty good at finding the good and the bad places now, and we're working across medical specialties at UC San Diego at the moment to define this map for many different diseases using exactly the same technologies and methodologies, we need to go a step further and develop more of a microbiome GPS that tells you not just where am I right now, but where do I want to go on this map, and what do I want to do turn by turn in order to get there. And ideally, we need to develop this kind of system and make it so easy to use that even our children can use it. So you could imagine a kind of smart toilet where as soon as you flush, it does an analysis of your microbiome and then delivers that report to your smartphone, which, let's face it, I bet you're using in there anyway. And by the way, we can tell when we sample your smartphone a lot about your lifestyle from that sample. And the idea is that it could show you in real time on the kind of microbiome map delivered to your smartphone, are you going in a good direction or in a bad direction? And is there anything in particular that you should be doing to stay in the good places of the map and maintain your health over a lifetime? But our ambitions are even grander than that, and this is the real science fiction part of the talk, although many of the pieces to do this exist, and a lot of the task is an integration task. But what we would like is we would like reading your microbiome not to be a bizarre thing that a few people do by sending it to the lab and having results come back weeks later, but rather to be as natural as examining yourself in the mirror in the morning. And so our vision is that as you breathe on that mirror, your breath will be whisked away through tiny pores in the glass and analyzed chemically. We're still working on the user interface for this, obviously, but displaying the chemistry in some user-friendly format is something we think is important. And before you laugh at this, remember there are already breath tests on the market for things like cystic fibrosis exacerbation and for SIBO and for other microbiome-linked conditions. And then we hope to take those breath profiles and translate them into a microbiome profile using some of the same artificial intelligence technology that backs Google Translate. Although it may be that that's impossible and we have to have your mirror communicate with your smart toilet over the internet to be able to retrieve your microbiome data. And then the idea is to integrate it into the types of map that I've showed you, which we've seen are very effective for communicating not just to scientists, but to physicians and to members of the general public about whether your location on that map puts you at risk for any particular disorder. And then perhaps gives you tips, again displayed in real time on that mirror as you're looking at yourself in the morning, about what you might try in order to guide yourself back into health. And while we're imagining things, we could imagine it communicating with your smartphone so that when you're faced with 1,000 yogurts in the supermarket, you can use augmented reality to identify the one on the shelf that you're looking for, pick it up and scan its barcode so that you're sure that you got the right one with the organism that you're looking for. And then you could have your smartphone with its activity tracker and with the information that it's collecting about your lifestyle and diet, communicate back to the mirror to modify your image in real time and show you visions of what you might look like in 5, 10, 20 years if you spent every day exactly as you spent today, or what you might look like if you did a little bit better or a little bit worse on those scores of activity, of nutrition, and of other things that affect the microbiome. So this might seem far-fetched, but almost all of those components exist somewhere. A large part of the challenge at the moment is putting them together into an integrated system and fitting the models to be able to understand how what you do today translates into healthy aging over your lifetime, which is a project that we're currently engaged in a large-scale collaboration with IBM at UCSD. So anyway, this is the kind of stuff that we're up to at the UCSD Center for Microbiome Innovation that I direct. A lot of what we're doing is putting together the expertise at different companies, so everything from the data acquisition end to the applications end to the data science that links them together, and also over 120 faculty members drawn from all across the university, from the College of Engineering, from the School of Medicine, even from the Scripps Institution of Oceanography, where a lot of the modelling people are, to be able to tackle these really difficult questions and to be able to do something that on a large scale will exploit the microbiome to improve people's health. So with that, I'd like to thank a large number of collaborators and funding sources, many of whom I mentioned during the talk with respect to their particular projects, the many members of my lab, both previously and currently, who it's been an amazing privilege to work with. Here's a snapshot of what the lab looked like about 6 months ago. As I mentioned, we couldn't do this kind of thing without a diverse range of different funding sources, including many government and non-government organisations, and tens of thousands of members of the public who have contributed directly to the American Gut Project. And finally, thanks for the invitation to come along to this wonderful symposium. I'm really looking forward to the rest of the talks and to the discussion, and I'd be delighted to answer any questions you have at this point. I'll just mention that I wrote a couple of books about these topics if you'd like to learn about them in more detail. The first one, Follow Your Gut, I know for sure has been translated into Japanese because I have a Japanese copy on my desk at UCSD. I think the other one is either in the process of being translated or is out now. But these will give you a lot more information about some of those topics if you're interested in learning more. So anyway, thanks again for your attention, and I'd be delighted to answer any questions you have.